라이카 현미경의 이호재 과장이라고 합니다. 제 목소리가 잘 들리기를 바랍니다. 일단 많은 분이 참석해 주셨는데 굉장히 이제 감사드리고, 오늘은 저희가 해마다 행사를 했지만은 아시다시피 이제 코로나가 지금 심각한 상태이기 때문에 온라인으로 부득이하게 하게 되었습니다. 하지만 많은 분들 참석해 주셔서 굉장히 감사드리고, 라이카 현미경은 우리 회사가 1849년부터 굉장히 오래된 역사를 가지고 있어요. 그래서 지금 올해까지 해서 171년 역사를 가지고 있으며, 지금까지 우리는 기존의 이제 현미경들이 할 수 있는 그런 뭐 이쁜 이미지를 촬영한다거나 그런 것들을, 그런 한계점을 밀어내고 새로운 거, 항상 혁신을 향해서 우리가 다가가고 있습니다. 그래서 많은 현미경 회사가 있겠지만 라이카가 새로운 기술, 새로운 장비를 만들었다 이렇게 하면은 앞으로는 이제 어떠한 형태의 트렌드가 이루어질 것이고, 어떠한 형태로 우리 기술들이 이제 뭐 진행이 되겠구나 하는 걸 가늠해 보실 수 있는 그런 길잡이라고도 생각하시면 좋겠습니다. 그래서 오늘은 그 혁신적인 제품들 중에 신기술이 이제 총 망라되어 있는 공초점 현미경 스텔라리스라고 하는 그 제품입니다. 그래서 단순히 이 제품이 뭐 좋다 이런 내용보다는 이 현미경이, 공초점 현미경이 기존의 라이카가 만들었던 것이라든지 기존에 있는 다른 회사들이 가지고 있는 그런 공초점 현미경하고 뭐가 다른지를 이제 오늘 소개를 시켜 드릴 겁니다. 그래서 가히 말씀드리면 굉장히 이제 다른 기술이라고 이제 보시면 될 것 같고, 하나씩 제가 좀 간략하게 먼저 설명을 드리겠습니다. 우선 이제 오늘 강의 중에서는 세 가지를 제일 집중해서 이제 들으시면 이해하시기가 좀 편할 겁니다. 3P라고 이제 보실 수 있는데, 하나는 파워, 좀 더 우리가 강력해졌고 좀 더 우리가 볼 수 있는 영역이 더 많아졌으며 짧은 시간 안에 많은 데이터를 만들어낼 수가 있습니다. 그래서 이 세 가지가 큰 카테고리라고 볼 수 있는데, 이 세 가지를 유념을 하시면서 어떤 기술 때문에 좀 더 강하고 포텐셜이고 프로덕티비티가 높아졌는지를 확인하시면 좋겠습니다. 우선 형광 현미경에서 혹은 공초점 현미경에서는 시료는 형광 시료를 사용을 하시죠. 그래서 이런 형광 물질들이 있는데, 이 형광 물질은 아시다시피 밤에 불만 끈다고 해서 반짝반짝 빛나는 건 아니죠. 어떤 특정한 빛을 조사를 해주면 여기서 어떤 이제 특정한 빛이 방출돼서 나오게 되죠. 예를 들어서 블루 계열의 빛을 조사를 하게 되면, 쏴주면 이제 여기서 반사돼서 나오는 빛, 에미션 되는 빛, 그린 컬러를 우리는 이제 눈으로 보시는 거죠. 그래서 형광현미경을 보시면 이제 재물대 위에는 분명히 파란색 빛이 나오고 있는데 눈으로 볼 때는 초록색 빛이 보이게 되는 거죠. 우리는 이 에미션 되는 빛만 보는 것이죠. 마찬가지로 그린 컬러의 빛을 조사를 해서 쏘아주게 되는데, 여기서 반응하고 나오는 빛, 에미션 되는 빛은 붉은색 빛을 이제 보는 것이죠. 그래서 여기서 그 중요한 개념은 익사이테이션과 이제 에미션인데, 이 현미경들은 이제 익사이테이션을 시킬 수 있는 거가 488이면 488, 555면 555 여러 가지가 있겠지만은, 튜너블로 이제 우리가 튜너블로 조정을 하실 수가 있어요. 488이면 488, 489면 489. 1나노미터 단위로 튜너블 해서 할 수 있는 점이 있고, 에미션 돼서 나오는 그런 영역도 우리가 튜너블로 조정을 할 수가 있습니다. 그래서 레인지가 굉장히 이제 다양한 레인지, 광범위한 레인지에서 우리가 자유롭게 조정할 수 있는 거는 지금 우리가 설명드리고 있는 우리 회사밖에 없고, 촬영되는 장치는 총 5개까지 꽂을 수가 있어요. 이 말은 5가지 컬러를 염색을 해도 동시에 촬영한다는 것입니다. 블루 하나 찍고 그린 하나 찍고 레드 하나 찍고 그런 식으로 진행되는 게 아니고, 총 촬영 장치가 5개가 있기 때문에 5가지 컬러를 동시에 촬영한다는 거죠. 그래서 수많은 G스택이라든지 타임랩스 이미징을 촬영할 때 우리는 굉장히 빨리 촬영할 수가 있는 게 신장비의 특징이라고 할 수가 있겠습니다. 다음은 신기술이 가지고 있는 거가 뭐가 있냐면, 우리가 보고자 하는 시그널은 여기 가운데 여기 있는 거예요. 여기 가운데 있는 것인데, 이쪽에 있는 이런 백그라운드 부분, 우리가 보고 싶지 않은 백그라운드가 있을 때는 굉장히 이제 방해가 되게 되는 것이죠. 그래서 이런 영역을만 보고 싶을 때 어떻게 해야 되느냐? 시료를 이제 많이 전처를 하시고 뭐 과산화수소 사용하시고 다양한 방법을 시도를 해 보시겠지만은 실제로는 많은 개선이 안 되는 것을 많이 경험하셨을 겁니다. 사실 이때는 어떻게 진행하는 게 제일 좋냐면, 타우라는 개념을 이제 적용을 해야 됩니다. 타우라는 개념을 적용해야 되는데, 타우가 뭔지 오늘도 같이 배우실 겁니다. 하나의 그 X축, Y축, G축, 그리고 타임, 요 정도의 그 1차원, 2차원, 3차원, 4차원까지는 이제 아실 거예요. 하지만 타우라는 말을 오늘 이제 계속 많이 듣게 되실 건데, 타우가 뭐냐면, 전에 파란색을 쏴주면 초록색을 보고 초록색을 쏴주면 파란색이 된다 그랬잖아요. 그래서 익사이테이션 시키고 수 나노 세컨드 뒤에 에미션이 되는데, 익사이테이션 시키고 에미션 되는 것이 그 시간이 조금씩 다 다릅니다. 시그널마다. 같은 알렉사 488과 GFP조차도 다른 것이죠. 그래서 어떤 시그널은 익사이테이션 시키자마자 에미션이 되고, 어떤 시그널은 익사이테이션 시키고 한참 있다가 이제 에미션이 됩니다. 그래서 전문 용어로는 포톤 얼라이벌 타임이라고 그렇게 되는데, 요 시간이 걸리는 시간을 이제 타우라고 이해를 하고 계시면 됩니다. 그래서 이 타우라는 값을 모든 이미지의 픽셀마다 다 다르게 적용을 하는 거예요. 백그라운드 부분의 타우 값은 색상으로 입혀서 짧다면 파란색, 그리고 길다면 빨간색으로 입히는 거죠. 지금 신기술의 신장비에서는 우리가 스위치 하나를 켜게 되면은 전체적으로 이렇게 색깔로 표시가 되게 되는 겁니다. 파란색은 금방 익사이테이션 시키자마자 에미션 된 거, 붉은색은 익사이테이션 시키고 수나노 세컨드 뒤에 에미션이 됐다는 것이죠. 그래서 우린 전체 이미지를 이제 염색을 특별한 핵을 염색을 했다거나 뭐 다른 부분 염색을 한 게 아니라 염색을 하지 않고서도 오토 플루오레센스만으로도 우리가 익사이테이션 에미션 되는 그 시간을 측정함으로써 이렇게 컨트라스트를 줄 수도 있고, 또 하나는 제가 초반에 설명드린 것처럼 오토 플루오레센스는 주로 이제 익사이테이션 시키자마자 에미션이 됩니다. 그래서 이런 시그널들을 배제를 시키고 트루 시그널들만 우리가 셀렉션해서 찾아낼 수도 있겠죠. 혹은 어쩌면 이런 것도 가능할지 모릅니다. 이제 알렉사 488과 M체리, GFP 같은 경우에도 타워 값이 다르기 때문에 같이 코스테이닝을 해도 우리가 세퍼레이션해서 우리가 이제 관찰할 수가 있는 것이죠. 혹은 간단하게 형광 염색을 하지 않고서도 이게 병리 시료입니다. 병리 시료에서도 우리 전체 픽셀의 디스트리뷰션이 어떻게 되는지를 봐서 알츠하이머의 플락이 어느 정도 되느냐 이런 것들을 타우를 이용해 가지고 이제 본다는 것이죠. 그러니까 기존에 그냥 익사이테이션 에미션에서 그린, 레드, 블루 이렇게 촬영하는 거는 기본이거니와 신기술은 이제 타우라는 기술이 적용이 되었습니다. 그래서 이런 타우 기술을 이용하면은 좀 더 이제 가까이 우리가 진실에 가까운 데이터를 확보를 하실 수가 있고, 이런 기술이 총망라되어 있는 제품이 이제 스텔라리스라고 하는 제품입니다. 그래서 이 제품에 대한 신기술이 굉장히 많이 들어가 있는데, 주 내용은 이제 타우, 그리고 제가 아까 설명드린 파워 포텐셜 프로덕티비티에 관련해 가지고 우리 이제 아담 클리프 선생님께서 발표를 해주실 겁니다.
안녕하세요, everyone. Thank you for inviting me back. This is now, I think, my third summer camp for Leica. It's a shame to not be in Korea this time. So thank you, Hojay, for the introduction. I'm going to talk to you. This talk is going to be in two different parts. I'm going to start off with an overview about our new Stellaris confocal. And something I thought would be interesting is at the end, I'm going to present actually an interesting paper that was done. Okay, sorry, Julia's asked me to turn back on my camera. Okay, so at the end, I'm going to talk a little bit about a paper that was done using our Falcon module, which is really the sort of currently the best-in-class fluorescence lifetime imaging system. And I thought this would just be interesting to present some examples of the kind of things that lifetime imaging can actually be used for. So let's start off with Stellaris. So there are three kind of things that are quite new about Stellaris. It gives you the power to see more out of your samples. We give you the potential to discover more, so to get extra layers of information. And we also, with Stellaris, make you much more productive. And again, this is just a nice example that Hojay also showed. This is an Arabidopsis root. And if we look at the traditional confocal image, you can see that with a single channel, we can see intensity, but we actually lose a lot of the information from our sample. If we look at the same sample on Stellaris, again, this is collecting a single confocal channel. One channel, nothing else. But if we take into account the extra information we can get from Stellaris, you can see, if I just use my pointer, you can see that there are actually 3 different kinds of fluorescence in this sample. So using Stellaris, we can discover much more about your sample without really changing very much about the imaging process. So our new confocal system is called Stellaris. This is a picture of it here. It's actually 2 different confocal platforms, the Stellaris 5 and the Stellaris 8. And now I'm just going to go through what for me are the most interesting new features of this system. So the first thing, and this is really the most fundamental, is the power that the system gives you. And in this case, it's not necessarily laser power, but we have a lot of technological improvements that help you get more information out of your sample. So the first thing that we've changed in the new system is we've introduced a completely new kind of detector technology. And the aim here was to give you brighter samples, so to be able to collect more light from your sample. This is an example. This is a standard 4-color sample, and this was imaged using a traditional metal alkaline PMT. So something that has been the standard for confocal imaging for a long time. If we look at the same sample using Stellaris with our new detectors, you can see immediately we get a lot more light out of our sample. For confocal imaging, initially you may think that this doesn't matter so much because I can just use very bright dyes. I can put more labeling on my sample. So maybe it's not so important to have a system that is sensitive. But really for confocal, the number of photons you collect is the most important factor for how you do your experiments. If you can get 15% more light or 20% more light out of your sample, then you can get the same image much faster. So we could go faster, we could collect more pixels, or we could decrease the laser power and get less photo bleaching on our sample. So starting off with more photons gives you much more flexibility with how you can then do any of your imaging experiments afterwards. So the new standard that we have for Stellaris is called the Power Heidi S, and the S is for silicon. So this new detector technology is a silicon-based multi-pixel photon detector, photon counter. So it's actually an array of little detectors instead of just being one. Each one of these has a silicon photocathode. And someone has asked to annotate on my slide. Okay. So we have a silicon photocathode and then a series of avalanche diodes under each one of these arrays. The important thing here is that this is not behaving like a camera. So the information that we collect from each one of these individual detectors is all combined together. So we're still using this as a single detector. We're not using this as a camera-based array or as a multi-pixel array. So this is still one detector that will give us the readout of 1 pixel. The nice thing about this detector is that we can actually use it in 2 different modes. So it can be used in an analog mode, and this is really how traditional PMTs or gas PMTs work on a confocal. The analog mode is nice. It gives you a very large dynamic range, so we can image very bright signal in the same frame as we can image very dark signal, and it gives us the highest photon detection efficiency. But we can also use this detector as a photon counting detector, and this means that we record individually when each photon arrives in each pixel. And this arrival time we can use, and we'll talk about this a bit later, we can use the photon counting mode and the arrival time to do some very interesting lifetime imaging experiments. So this is now our standard detector. We do not sell PMT detectors anymore on a confocal. And the reason for this is very simple, that this technology is so much more advanced than PMTs or gas PMTs that we didn't feel the need to continue selling PMT detectors. So the nice thing with this detector, again, as I said, we can use it in an analog detection mode, which gives us a very large dynamic range, but we can also use it to do photon counting. And the photon counting is very helpful when we combine this together with a white light laser. And the reason for this is the white light laser we used is pulsed. So we know exactly when the laser is emitting light pulses, and this is usually every 12.5 nanoseconds. So we know when the light hits the sample, and we can also then count exactly when the photons are received back in the detector. And this is one of the requirements we have for lifetime imaging. And we'll come back to this a bit later on in the presentation. The other thing that we've changed in our new Stellaris system is that we've changed, or we actually invented a new way of doing photon counting. And traditionally on systems, there are other ways of doing photon counting in imaging. And the idea is that you take the signal from your detector and you get a little trace, and then you record a peak and you say, okay, when I see a peak, this is 1 photon. And then you can imagine as if you have 2 photons arriving quite close together. In this case, we can still resolve the gap between the peaks. So I can still say this is now 2 photons, not 1. But on traditional photon counting systems, if the 2 photons get much closer together, at some point it becomes impossible to count them as 2 photons anymore. So on most systems that use photon counting, the drawback is that when you have very bright samples with a lot of light, you often start miscounting. So you will record 2 photons, but you count them as 1. The new power counting technology that we've patented allows us to measure single photons. We can look at 2 photons together. And also when these 2 photons get very close together, we can actually still discriminate this using some signal processing. So we can say this is definitely 2 photons. The advantage for this when you're imaging is that, again, this gives us a lot of precision when we're doing lifetime measurements, but it also means if you're quantifying any fluorescence, we can do incredibly accurate quantification at very high light levels. And this is something that we've now patented and is now built into all of our systems. The main improvement of the new detector is the photon detection efficiency. And I should say now that photon detection efficiency is very similar to quantum efficiency, which is the normal way that people measure gasp detector sensitivity. But actually, photon detection efficiency is a much better measure of a detector because it's the measure of the total probability of when you have a photon hitting the detector, what is the probability that I measure that as an electron or measure that as count that as a signal? So quantum efficiency for detectors actually only measures the very first step, which is the conversion of a photon to an electron at the front of a detector. So I'm in the talk, I'm just going to talk about photon detection efficiency. This is always at least the same as quantum efficiency or often a bit lower. So if I say the PDE or the photon detection efficiency is about 60%, this is probably actually an underestimate of what it would be compared to a gasp detector. So the really important thing with our new detectors is that we have a much larger sensitivity, especially in the blue and the green range. So this is a comparison. This curve here would be a normal gas PMT on a regular confocal, and we get at least 15% more signal or better detection efficiency in the blue and the green range. And this is typically the range where most people do live imaging. So we're imaging with GFP, maybe with cyan fluorescent proteins. So we have a very big improvement in sensitivity in this range. We can still detect red dyes very nicely, but the other very big difference is that these detectors are very sensitive longer than 750 nanometers. Someone has just raised their hand. I don't know if that's... Okay, I'll keep going unless someone tells me to stop. So the other advantage of this detector is they're much more sensitive into the near infrared range. And this is actually a very big problem with traditional gas PMTs, that they are quite sensitive for blue dyes, green dyes, red dyes, but the detection sensitivity drops to pretty much 0 at 750 nanometers. So in reality, the longest wavelength dye you can use would be Alexa 647. You can buy a confocal with a laser that will let you excite dyes like with a 700 nanometer laser, but it's very hard to get any sensitivity in that range. Using the new detectors, we have a much better sensitivity into the far red range. So we can use dyes like Alexa 750 or Alexa 700, and I'll show you some examples of that later. On the Stellaris 8 system, again, the HIDI-S is our standard detector, but we have a couple of other detectors that have slightly different performance. We have the HIDE-X, which is based on our current hybrid detector technology. And this is really intended for doing FCS and very high precision STED experiments or FLIM experiments. And the other detector, which is, again, very nice for people doing live imaging using near infrared dyes, is the HIDE-R. And this has a much higher detection efficiency in the far red range. So depending on the kind of applications you need, we have different detector options. But for most people's applications, the HIDE-S is much better than anything else around, which is really nice. So the first big change we have is something that we give you more sensitivity. We can detect more signals and we can see much lower levels of light on your sample. The second thing we've done is actually increase the number of colors that you can use as standard on a confocal. And at least for me, when I used a confocal in the past, I typically did 4-color experiments. So I would use DAPI, I would use Alexa 488 as a green dye, Alexa 568 as a red dye, and maybe Alexa 647 as a far red dye. And that was based on the sensitivity of the confocal system and the lasers that are available. With Stellaris, we can actually add into the far red range, we can add another 2 labels. So this is just an example. On the left, we have a cell that's labeled with a far red dye, and with Stellaris, we add 2 more channels to that signal. So just to show you the difference, this is typically where the detection would stop on most confocals with Alexa 647. And on the sample on the left, we're using Alexa 647, Alexa 700, and Alexa 750. So, or 790. So in this case, we can easily add 2 or 3 more labels to your samples just by expanding the spectral range. If you think about how you do experiments, this actually saves you a huge amount of time. Traditionally, if you were staining a brain, like a tissue section, like a brain slice, if you can only label 4 things, so you have 4 colors you can use. The first one is normally DAPI. Okay, so then you have 3 colors. And then if you're taking this from a mouse, which is expressing GFP, okay, now you only have 2 colors left. So if I wanted to label 3 or 4 different markers in my tissue, I would have to make several different tissue sections, and then the first section would be labeled with DAPI and GFP, and then maybe a cell division marker and maybe a blood vessel marker. Then I would take another section and I label with DAPI and GFP, and then the red dye I would have to use for a hypoxia marker, and the 4th one I would use for actin. So you end up making many, many sections and then trying to compare these results. With Stellaris, we can actually label 5 or 6, or up to 7 or 8 colors on the same sample. And this means that with one tissue section, I can get much more information, and it's much easier to correlate and look at the co-localization of different molecules. So there are a couple of ways we do this. Firstly, we've expanded the range that we can tune our detectors, which we talked about earlier. We have detectors that are very sensitive into the far red range, but also we've changed, or we're using mainly white light lasers to do the excitation. And on an old confocal, you have fixed wavelength lasers. So we have maybe a line at 405, we have one at 488, we have something at 560, and maybe a 630 line. And those fixed laser lines usually determine which dyes you can use well. On the Stellaris 5, we keep the 405 line. We can have a 448 line for doing CFP, but then all of the other excitation wavelengths are covered by the white light laser. And if you've never seen one of these before, it's a very different way of working. The laser can produce laser lines at any wavelength within this range, and we can make up to 8 lines at once. So I can very quickly just pick the absolute best wavelengths for the dye I'm using. And this has quite a big impact on all of your imaging. As a very simple example, if you were using Alexa 488, which is one of the most standard dyes for one of the most standard green dyes, if you use a normal confocal, then you excite this with 488 nanometer light. And it turns out that is actually not the best wavelength to excite this dye. The best wavelength to excite Alexa 488 is about 498 nanometers. So if I go 10 nanometers longer, I get about 10 or 15% more signal out of my dye for the same amount of light that I put on it. And if you think about this as an example, this means that we can get much less photo bleaching. It's even more powerful for live imaging where if we're working with fluorescent proteins like M-cherry, with M-cherry on a normal confocal, we would have to try and excite it at about 560 nanometers. And that will excite M-cherry, but it doesn't excite it very well. The best excitation would be about 595. And if I tune the laser to this wavelength, I can use half the laser power and still get very nice, or get exactly the same level of fluorescence. So for live imaging especially, if I can optimize the excitation to the fluorescent proteins I'm using, I can use much less laser power. And if I use less laser power, it means I get less photo damage of my cells. So I can be much more sure that my cells are happy during my experiment. I'm actually making real videos of live cell imaging. I'm not making videos of cells dying. So the white light laser is incredibly powerful. On the Stellaris 8, we have a much bigger version. These are the third generation of white light lasers that we've used. And on the Stellaris 8, we have a laser that can image from 440 to 790 nanometers. And this really covers a huge range of dyes. But even with the Stellaris 5, we can image most common far red dyes. We can excite Alexa 700 or Alexa 750. So this is just some examples. Even with the Stellaris 5 laser, we can image and excite a lot of these dyes, and we have a system that is sensitive enough that we can detect them. One of the reasons that this works, and if you've used Leica confocals a lot, it's very difficult to go back to an old-fashioned confocal because most old systems use dichroic mirrors. And the idea with the dichroic mirror is that we have the laser coming into the confocal, and the dichroic mirror reflects the laser into the objective. Then the fluorescence that comes back from the sample goes back and it passes through the dichroic mirror and it goes to the detectors. And this is how every other confocal on the market splits the lasers and introduces the lasers into the light. So using the dichroic mirrors, we can use, we're very limited in the combinations of lasers we can use. On the Leica systems, we use what's called an AOBS, which is an acousto-optic beam splitter. And the way this works is that we shine the laser light through this crystal, and if we do nothing, the laser just goes straight through and it hits the back of the scan head, so nothing happens. If we apply a particular radio frequency to this crystal, then the laser light is bent into the right angle and it gets directed to the objective. Any light that is slightly different wavelength will just pass directly back through the crystal. So any fluorescence that comes from the sample just passes through the crystal and goes to the detector. And we can introduce multiple different laser lines at once. The benefit of this is that the switching takes microseconds. So we can switch very quickly between lasers, and we don't have to keep changing the dichroic mirrors that we're using in our system, which is a very slow part of live imaging. The other really big benefit is that if you look at the way that dichroic mirrors work, you actually lose quite large amounts of fluorescence on either side of the laser line. So if we're using this is a standard three color dichroic mirror, which we would use if we have 3 colors in our imaging setup. The problem is that if we're using a 561 nanometer laser, we will lose about 30 nanometers around that laser line. And if you imagine that the fluorescence of GFP would be something like this, and actually I can show this on the annotation. So if I just annotate this on here. So GFP looks something like this. And if we're using a normal dichroic mirror, we're actually losing this big chunk of your GFP fluorescence. With the AOBS, we lose about 1 nanometer each side of the laser line. So we get much more efficient collection, and we just collect more light when we're imaging. So for live imaging experiments, this is another area where we can just gain more photons and get more light out of our sample. For most users, the best part is that you don't have to think about any of this. You just choose the laser line you want, and the system does everything else. You won't get weird messages that you have the wrong mirrors, or you won't try and spend a long time working out why your experiment isn't working. So that's the sort of the main changes in the hardware. Next, let's look at what we can do, which is really using some more new technology. And the techniques that I'll introduce here are collectively called TauSense, and this covers really 3 different approaches. So the idea with TauSense is that instead of just looking at the intensity of the fluorescence we collect, so how many photons do we get, we also take the arrival time of each photon into account. And this gives you an extra dimension of information. So it's in the same way that on an old confocal we would look at different colors of dyes, we can look at colors in one dimension and we can look at the arrival times of the photons in another dimension. And a nice example of this is tau contrast. So again, this is the example we presented at the beginning. These images were both taken at the same time with a single detector. And using tau contrast, I can get much more information out of my sample. And this is all based on fluorescence lifetime. So just as a very quick summary, if you have a fluorescent dye, if we excite this with a photon, the photon is absorbed by the dye. This causes an electron to be excited. The electron spends a little bit of time in the excited state. It loses some energy by vibration, and then it drops back down to the ground state. And when it drops back down, the fluorophore releases a photon of light, which is a longer wavelength. The important part here is that this process takes time. And for different fluorophores, this process takes different amounts of time. So as a nice example, if you took Alexa 555 and Alexa 568, they both go through the same process. We excite them both with green light and we get red emission from both dyes. But Alexa 555 takes on average about half a nanosecond to do this cycle. Alexa 568 takes about 2 nanoseconds to do this. So we can get, if you can measure when the photons arrive, you can actually start to use that information to separate these 2 dyes. And the reason we can do this is that we have a pulsed laser, so it's very important that you know when you hit your dye with a photon, and the white light lasers we used are all pulsed. And we have very sensitive detectors that can measure exactly when the photons arrive. And the idea here is that we put all this information together and we collect something that is just called the average arrival time. And this is actually all calculated on the scanner. So you don't actually need to look at the data. In the case of TauSense, we don't record the arrival times of individual photons. We just get a number for each pixel. So all of this is done by the confocal. It's actually incredibly easy to do. So in this case, if I wanted a tau contrast image, I can take my setup in the confocal window and I just turn on tau contrast. So I flick a switch and I get an extra image of information. We still always record the original confocal image, but we can record the tau contrast image at the same time. So just as an example, and this is the same one that Jose showed earlier, this is the standard fluorescence intensity image. And if I turn on tau contrast, I automatically get this image as well, which is now showing color coding the image based on the average arrival time. So blue would be short arrival time, red would be long arrival time. And even within this very small, ugly image, you can see there is a big difference between the nucleus here and this part of the cell over here. So tau contrast is a very powerful technique. We can actually use this as well as looking at autofluorescence, and this is one example. We can use this for things like FRET experiments. So we can do fluorescence lifetime imaging. We can look at dye molecules that are easily, that we know change lifetime based on the environment. So for example, Nile red is a very standard fluorescent dye. This binds to lipids, and depending on the lipid composition, you will get a different lifetime in the plasma membrane compared to the mitochondria. Sorry, someone has just asked, what is the pulse laser? So the white light lasers we use typically pulse at 80 megahertz. So it's about 1 pulse every 12.5 nanoseconds. On the Stellaris 8, we have the option of changing the pulse repetition rate so we can go much slower. But typically for a lot of biological dyes, 12.5 nanoseconds is quite a good time because the lifetimes are between 0.5 and maybe 4 nanoseconds. So this is usable. Okay, so we can use tau contrast to do, for example, FRET experiments or we can do fluorescence lifetime FRET experiments, which is the most robust way of measuring FRET, and we can look at these different biosensors. Another option we have is tailgating, and again, as Jose mentioned, we can use tailgating to get rid of some signal that we don't want. So in the case of the image on the left, there is some reflection from the cover slip here, and reflection has very short lifetime. The lifetime is essentially 0. So with tailgating, we can just say ignore the photons that arrive within the first half a nanosecond after each pulse. And then we're still left with the rest of the fluorescence, and we end up with nice images, but with much less background. Using tailgating, instead of just throwing the data away, which is what we could do on the SP8, with tailgating, we can actually then separate this information into another channel. So we can actually keep it. And there are also some clever tricks you can do with this in STED imaging, for example. So that's tailgating. It's a very powerful technique, but the one that I think is most interesting is, and I'll skip over this just to save some time. So this is just an example with tailgating. So the nice thing now is we can collect both images. So this is the first gate. This is all of the arrival time. So essentially the original image. And the second one is just the gated image, so removing this non-specific signal. This is my favorite tool of the three. This is tau separation. And in this case, we're using the average arrival time and the lifetime information to completely unmix dyes. And this is an example. So this is a 2-color live cell imaging experiment done on a traditional confocal. So here we have set one detector to collect green signal and the other detector to collect far red. And the difference between a traditional confocal and what we can get on Stellaris is that in this case, we're using 4 dyes at the same time. And the way that this works is that we can separate dyes based on lifetime. So in this case, in the original image, we have one channel collecting the yellow signal. So this is collecting around the green range. The white channel is collecting in the far red range. What you see when you look at the tau separation is actually the green channel is we're using M-neon green, which is a green fluorescent protein. And we're also using mitotracker. And mitotracker is a very standard mitochondrial dye. So the mitotracker is shown in green here. The M-neon green, which is fused to life actin, is showing actin labeled in red. So we have 2 very standard dyes imaged in one detector channel. In the far red channel, we actually also split this up. So we're using silicon rhodamine tubulin, and this is a very nice dye from spirochrome. You can add this to live cells, and it gives very nice labeling of tubulin, which we see in purple. But also on this sample, we've added NUC red, which is shown in blue, so this kind of blue cyan color. And NUC red is essentially Alexis 647, and this, again, has a very different lifetime, and this labels nuclei. So here, using 2 channels, we've managed to collect 4 dyes at the same time. So this is the other powerful thing we can do with Stellaris. I can add more colors just by using a broader range of the spectrum, but then potentially I could put 2 or 3 different colors into each channel and use the differences in lifetime to separate them. So again, all of this we've designed this to be built into the software. So if you want to do tau separation, you just choose tau separation as the mode. You can open this little preview window and you can see the distribution of the 2 lifetimes. And if you just press find, then the system will work out the best distribution or the best separation for you. You can also adjust this manually if you like. But all of this is calculated on the confocal or in the confocal hardware while we're acquiring the image. So once you've set it up, you just capture your images like a normal confocal. And at the end, you just get 4 confocal channels or 4 normal images out of your data, where on a normal system, you would have 2. So we have these 3 different modes of tau sense. Tau contrast is very powerful when you're looking at autofluorescence, and it's also very good for doing things like FRET experiments. And anything where you want to see if something changes over time, this is a very powerful technique for a lot of live imaging. It's also a very nice way of looking in your sample to see if there really is any big differences in lifetime for a particular channel. And you may discover that, and we discovered this last year at the summer camp when we were looking at the Alzheimer's brain sections, which Jose showed earlier. And we found that actually you can see these very big differences in lifetime in these Alzheimer's plaques that you don't see in a lot of the background staining. So tailgating is a nice way of removing some of the non-specific signal, things like reflection and very short lifetime autofluorescence. And tau separation gives us a really powerful way of separating dyes. Or we could potentially separate a fluorescent dye from autofluorescence in the tissue. The last bit of the new stuff about Stellaris is the productivity. And the idea here, again, as Jose showed earlier, is to make the system as easy as possible to use. So the first big improvement we have is this setup called Image Compass. And the idea here is that it makes the whole system very intuitive. So you can see just by looking at this display, we are doing 3 different sequences in our acquisition. And in the first sequence we're collecting a green and a far red dye. The second sequence, we're collecting a red and a near infrared dye. And the third one, we're collecting DAPI. And the idea here is that you can just use the dye database. You can drag and drop dyes into the display. And then you can press start and you can very quickly set up the optimum. In this case, this is a 5-color sequential scan, but you can get the best imaging settings just by dragging dyes around. This is really helpful if you're new to confocal, because this typically, how to set up the system is one of the hardest things to learn when you first start. And actually, if you go into the software, if I click on a particular channel, it will give me an estimate of how much crosstalk we have between the different dyes. So you can also still go back and use the software in the way that we've used before. I'm quite old-fashioned. I like moving the detectors around on my own, and I like adding laser lines myself and tuning the detector. And just as a comparison, this is a race between the 2 approaches. So this is, on the left, this is how I would normally set it up. And on the right, this is using the simple dye-based approach. So if we just start the experiment, the aim here was to set up a 2-color experiment. So in this case, we've just made 2 different acquisition sequences, and the new user has already finished. So they just selected 2 dyes and dragged them into the system. For the high-tech user, so me, this person is still trying to move the laser lines around, optimize the detectors, and the whole process takes a lot longer. So at the end of this experiment, in 10 seconds, I can set up the confocal for imaging, or I can go and adjust it myself, and I will still get the same results, but it takes me 3 times longer. So the idea here is that we make it very easy for users. We also, on all of the channels, on the images you're looking at, we make it very easy to identify which dye you're using in which channel and which detector. And you can control this very easily through the panel box, which is this manual dial device that we can use to very quickly adjust each channel. The other thing that we've done is made the system very fast. So we have the option of a resonance scanner, which lets us image at very high speed. And we can combine this together with our Lightning confocal super resolution approach. And this lets us get very fast imaging at very high resolution with very high quality. And this is combined together with a new rolling average approach, which we've called dynamic signal enhancement. And the idea here is that using this, we can capture very fast time lapses, and we can still average across the time lapse and get a very powerful result. So this is just an example. Hopefully this will stream nicely on the video. This is a migrating lateral line, or a group of lateral line cells migrating in the zebrafish. This was taken with, I think, 5 volumes a second, and the total time is about 1.5 hours. So we're doing a very fast set of Z stacks, and in this case, we're using GFP and mcherry. And the nice thing here is that I think there's 150 stacks in total, and you don't see any obvious photo bleaching of the M-cherry in the process, which is really, really impressive. So one of the nice things in our new software is that we can switch between the normal scanner and the resonance scanner just by pressing a button. So I can very quickly switch from I can switch to fast scanning to do an overview. Then I can switch back to slow scanning if I want to get more detail. And when you combine the resonance scanner together with the dynamic signal enhancement, you can see with the resonance scanner images, this is a live cell being imaged at 50 frames a second. We're collecting very small numbers of photons. So if I just start the video again, you can see that it looks a little bit noisy. We're collecting maybe 5 or 10 photons per pixel at most. If we apply the dynamic signal enhancement, we have the same time resolution. So we're still imaging at 50 frames a second, but we can get a much clearer image. So a lot more information out of our sample. And the wonderful thing with this technology is that we can use this either while we're acquiring live, or we keep the original data. So you can go back afterwards and reprocess your image and adjust the different parameters and make sure that you really have the best averaging and the best smoothing for the time resolution and the speed that your sample is moving. Lastly, we also have our Navigator view, and this I know doesn't play so well over the system, but this is just a way of this is a bit like Google Maps or Google Earth for microscopes. So we can very easily set up very large tile scans. This is a 3D tile scan using tau contrast of a plant. It's actually part of a flower. You can see the pollen grains have a lot of interesting lifetime. This was recording a single channel and then also recording the tau contrast at the same time. So using Navigator, we can image very large tissue sections, and we can do very efficient multi-position and multi-well acquisition. And all of these things fit together very nicely. So we can use the resonance scanner to go very fast. The new rolling average function works very nicely together with Lightning. And we can also use Navigator together with this. So all of these things work synergized together very easily. So as a little summary, we have two systems. The Stellaris 5 is really intended as the standard confocal for most labs, and the Stellaris 8 has a bit more upgradability for core facilities. So the Stellaris 8 has the bigger white light laser, and we have the option of upgrading it to multi-photon or STED or Falcon, which is our really high-end FLIM, very precise FLIM system. So that's the quick overview of Stellaris. In the last few minutes, I just want to give you a very nice example of how fluorescence lifetime was used by a group in Oxford to study HIV infection. And in this example, so this is a group of Sergei Padilla-Para from Oxford. He is interested in a lot of different imaging techniques, but in this case, they were imaging HIV infection. And HIV is quite a difficult virus to image. It starts off as an immature virus. So all of the different proteins are packed into the virus, and then there's some processing, and the virus really assembles and matures into the fully functional infective virus. Imaging single virus particles is normally very difficult. There are not huge numbers of protein molecules within a virus, so it's very hard to label them with a lot of GFP molecules. Often, if you make GFP fusions of a particular protein, that will block the function of it. So it's very easy to make a GFP-labeled virus, but it's very hard to make a GFP-labeled virus that actually works and behaves like a normal virus. And in this case, there are a couple of groups that have found different ways of labeling two of the different proteins, and we'll talk about examples. But the other problem is that traditionally, or typically when you do this, you have very small numbers of fluorophores per particle. So in this paper, one of the main fusions they had was fused to the GAG protein, which is one of the main components, and they found that to make the virus functional or viable, they could only have about half of the GAG molecules labeled with GFP. If all of them were labeled, then the virus wouldn't infect. So they have very low levels of GFP expression, and you need a very sensitive confocal to be able to track individual virus particles moving around and track them fusing with cells. So if you're interested, I'll put this up again at the end. This paper was published in PLOS One very, very recently. Oh, sorry, this is the original papers on the viral fusion, and I'll give you the paper where they published this. So there were a couple of different modes they used. So the interesting thing in this paper is they started with a question, and the question was just based on an observation. If you have a dish full of cells and you infect them with a large number of virus particles, you don't see all of the cells being infected. What you tend to see is patches of cells. So some cells will become infected, other cells will not be infected. And in an animal, you could understand this, but in a tissue culture dish, this was very confusing to them because you would expect all of the cells would be very similar. Genetically, they should be the same, but they still had this case where they would see some patches of cells being infected and others not. In this case, they're using a fluorescence lifetime imaging probe, so any of the red cells here are cells where the virus has successfully fused with a cell and infected it. So there could be two reasons for this. Either some cells are much more sensitive to infection, or some cells are more resistant to infection. And one of the things they thought that might affect how well cells are, how likely cells are to be infected, is metabolism. And what they did in this case is they added a metabolic inhibitor, so they added 2-deoxyglucose. This slows down the metabolic function of the cells. And what they could see is that in untreated cells, they get 100% of the cells will become infected over time. As they start adding 2-deoxyglucose, suddenly you start seeing a decrease in infection. So they had this observation that if you block them, if you slightly inhibit the metabolism of a cell, you get less viral infection. And the next question then is that a cause or an effect? So if I can see an individual, if I see two cells, if I know one of them has a very high metabolism, one has a low metabolism, and I see virus particles fusing with both, can I tell whether they're infecting or not? So they needed a few different tools to be able to do this. So they needed some kind of biosensor to look at the metabolic state of a cell. And in this case, in the previous graph, they were looking at all of the cells in a dish. But it's very hard to tell the metabolic state of each cell before it's infected. So they want a sensor that will let them image the metabolism, and they also want some way of fluorescently labeling a virus particle. So, and ideally, they would like to measure some assay to tell whether the virus particle has really fused with the cell or not. Because even if the virus can't fuse, it may just stick to the surface. So the way that they did this is they grew their cells on these little marked gridded cover slips from Ibidi, so they can very easily find out which of the chambers they were growing, or which of the chambers the cells, or exactly where the cells are. They can go and find the same cells later. They have a FRET-based probe for ADP, ATP level called Percival, and this gives you a change in fluorescence lifetime depending on the amount of ATP in the cell. They used fluorescently labeled virus particles, and also they used another assay, which is a Beta-Lactamase assay. So this is a fluorescent dye that, when it is cleaved by the Beta-Lactamase protein, changes color. And they can use HIV virus particles that have a small amount of Beta-Lactamase inside the virus. So if the virus particle successfully fuses with the cell, the Beta-Lactamase enters the cell, and then it cleaves this dye. So just to show you what this assay looks like, in this case, initially the cells are green, and then any cells that, as soon as the cell becomes infected with the virus, it immediately turns red. And they've just pseudo-colored this. So you can see now that if we look at lower viral levels, you can see with just virus alone, we get a mix of infected and non-infected cells at a particular time point. And then as we lower the cell metabolism, you can see that fewer and fewer cells are being infected. So they have this very nice readout of whether the cell has been infected. They can image single viral particles, and they can also measure the metabolic activity of the cell before the virus is added. So what they found in this case is that as you block the metabolic activity, you decrease the ability of the virus to fuse with the cell. So what they wanted to know next is, okay, we know if the metabolism is low, the virus can't infect the cells very well. But what stage of the fusion process is this being blocked? And they used a very clever experiment to do this. So in this case, they set up an experiment using a 2-color virus. So they took the virus particles, they labeled the membrane of the virus with DID, which is a red lipid dye. And they labeled the GAG protein, so one of the cytosolic proteins of the virus, with GFP. So when you look at these individual virus particles, you see little yellow dots. You see green and yellow. And you can see these little dots diffusing around the media, and then at some point the dot will bind to the surface of the cell. And once it's bound, you will see a little yellow dot sitting on the membrane. What happens next is a step called hemifusion. So the membrane of the virus actually fuses with the plasma membrane of the cell. And if the virus gets stuck at this position, what actually happens is that the lipid dye in the membrane can diffuse into the plasma membrane. So you see a yellow dot, and as soon as the virus membrane fuses with the plasma membrane, the red dye disappears. It just diffuses across the whole plasma membrane, and you can't see it anymore. But because the whole fusion hasn't taken place, the green fusion protein in the middle will still stay there. So you know that if you'll go from a yellow dot to a green dot, this means that the virus has tried to fuse with the cell, but the soluble proteins inside haven't been released into the cytoplasm. And there are actually 2 different things that could happen here. So either you could then have full fusion with the plasma membrane, and then all of the GFP proteins would be released into the cytoplasm, and the green dot would also disappear. The other possibility is that before this diffusion event happens, the whole virus particle is endocytosed. And if you look at the endocytosis, what will then happen is the red dye will diffuse around the endosome, but it won't be able to go anywhere. So you'll still see a red dot. But the virus particle can still fuse with the endosome and release the GFP into the cell. So if you see only red dots, you know that the virus particle has been internalized, and then it's managed to actually successfully fuse. And this is actually what they see when they look in the control cells. They see that most of the time the virus particles get endocytosed very quickly, and then they fuse with the endosome and they release the virus particle. They release all of the viral proteins into the cytoplasm. So you see the green signal drops and then it suddenly disappears. The red signal stays about the same. So you can see these little yellow dots, and over time they become red dots. So we lose the GFP from inside the virus, but the red is confined within the endosome. If you decrease the metabolism of the cell, then what you see is the opposite. So you can see these little yellow particles attaching to the cell, and then we lose the red signal. And we lose the red signal because the virus is stuck at the plasma membrane, and because it's stuck at the plasma membrane, the red dye can diffuse away, but the green is stuck inside the virus. So the nice thing is they could look at why the cells are why the virus can fuse with some cells and not with others. Then they can actually go into the mechanism and really dissect where in the mechanism is there a problem. And this is just another example. So in this case, what they then went to think about is, if you lower the metabolism of a cell, why would that stop the virus from infecting? And one of the things that people know is that the metabolism of a cell will also change the lipid composition of the membrane, and it changes how much tension there is in the plasma membrane. So in this case, they're using a different marker, which is a GFP labeled virus, an M-cherry labeled virus, which is in red, and they're using a marker for membrane tension. And this is another FRET probe, which they measured using FLIM. So you can see that the green here is marking the plasma membrane of the cell. And you can see when the virus gets very close, and you can actually see the fusion event. So you can see the virus is here, and then suddenly it fuses and the M-cherry enters the cell. The interesting thing with this probe is that if the membrane is under a lot of tension, it will pull these 2 fluorescent proteins apart, and you will see a FRET change. So what you can do is you can actually say, if I see an increase in lifetime, it means there is more membrane tension. So you can look at how much tension there is during the different parts of the process. This is a slightly confusing graph, but if you just focus on the green line to begin with, so here they were measuring the tension at the point where the virus particle is on the cell. And you can see initially it's quite stable, and then suddenly when you have fusion, you see a drop in membrane tension. And then the membrane will recover, and then everything stays there. If you have no virus particle, the tension stays at a fairly similar level. At the top, if you decrease the metabolism of the cell, you can see the point where the virus is sitting there, but you don't see this drop in membrane tension. So you don't see viral fusion successfully. Interestingly, you can rescue this effect by adding more cholesterol to the cells. So if you decrease the metabolism but add some more cholesterol, you see this drop in membrane tension again. And actually the virus particles can go and infect the cells again. So this is just a nice example. Here we were taking fluorescence lifetime imaging, so data we can get from FLIM, correlating this with very high resolution single viral particle tracking, and adding this with some interesting virology and interesting cell biology to get a very nice story. And this is this is the reference to the paper here. I'd suggest you go and read it if you're interested. It's a very nice publication. And this is something that it's really only possible to do with our Falcon FLIM system. And this is something that we have on an option on the Stellaris 8 system. So using Falcon, we can do these very high speed, very high precision FLIM measurements. We can tune the excitation and the emission of the system to make it very easy to add multiple labels at the same time. So in most of the figures of this paper, they were looking at fluorescence lifetime and looking at FLIM threat, but also tracking the virus particles or looking at the infection reporters. So it's very interesting to do this, and all of this is integrated very nicely with the confocal functionality. So this is from a different group, but this is just an example. It's very easy to do this in 3D. So this is a nice example of 3D FLIM threat on a group of cells. And that's the end of my presentation. Thank you all for listening. Hopefully that wasn't too long and you managed to keep up with me. If you have any questions, please ask